Hello, my name is Richa Nathan, and my research project has been on the incidence of the leptin receptor mutation Q223R in obese pediatric patients in rural East Tennessee. My mentor has been Dr. Jason Abercrombie at the Webb School of Knoxville.
So what this mutation is, it's associated with childhood obesity. Okay, so I'd like to start off with some background. So how does one define childhood obesity? Today, according to the CDC, we define a child as either overweight or obese based on their BMI, which is their body mass index. And this is just a simple calculation with their height and weight in proportion to the other kids their age and the same sex as them. So if a child has a BMI from greater than the 85th percentile or to the 94th percentile, they're considered overweight. And if they have a BMI greater than the 95th percentile, they're considered obese. What causes childhood obesity? There are a multitude of factors that can cause childhood obesity, including but not limited to socioeconomic factors and nutritional factors. But in Tennessee, the main ones are a very high calorie diet with a lot of sugars and processed foods, and sedentary habits, which is just inactivity. But in addition, today we are seeing a lot of more research in the literature that's proving that genetic mutations are also associated with childhood obesity. And that's where my project is more geared towards. I was curious to see if LEPR, this specific mutation, is truly influential and is one of the factors in childhood obesity. So the next point is, what are the long-term impacts of childhood obesity? There are also a multitude of impacts that can be seen later on in the child's life. But the main ones, especially here in Tennessee, are hypertension, type 2 diabetes, and heart disease, all by the age of 25, which is extremely alarming because this is just a very young age, and that just proves that childhood obesity is extremely dangerous. It's a huge epidemic. In addition, an alarming fact is that Tennessee has the largest amount of kids with type 2 diabetes, and this is just crazy because people only develop type 2 diabetes by the age of 45 or older. So this leads me to the point of the extent of the epidemic here in Tennessee. So I'd like to refer to this map by the National Survey of Children's Health.
This map shows the combined overweight and obese rates for children ages 10 to 17 across the country. All the purple states, including Tennessee and Mississippi, Alabama, Florida, West Virginia, and North Dakota, have a greater than 35% rate. In Tennessee specifically, 38.5% of children are obese and overweight. This is an extremely alarming rate due to the fact that so many children are affected by this epidemic.
All of these facts led me to my research question and to think about the extent of how this epidemic truly affects my community, and I wanted to do research that would help my community, basically. So my question is there an incidence of leptin receptor deficiency in obese pediatric patients in Tennessee? So what is leptin receptor deficiency and how does it contribute to childhood obesity? So leptin is a hormone that flows in our bloodstream in accordance with the endocrine system, and it flows in proportion with our body fat mass. So if one is very obese or overweight, they have very high levels of leptin in their bloodstream. But leptin has the job of controlling our eating levels and our energy expenditure, but it can only do so if it binds to the leptin receptor in our hypothalamus.
In this figure, the red molecule is leptin and the larger blue molecule is the leptin receptor, which is sitting in our hypothalamus. When leptin binds to the leptin receptor, which I've indicated with an arrow, this signals the transphosphorylation cascade named JAK2, which is indicated with the green spheres at the bottom. When JAK2 is signaled, this tells our brain that we either need to stop eating or start exercising, and it controls our energy expenditure. But when the mutation that I've been studying, named glutamine 223 arginine, occurs in the leptin receptor, which I've indicated with the arrow at the top, this completely changes the orientation of the leptin receptor, and leptin can no longer bind at the binding site, which I've indicated with the bracket at the leptin binding site. This binding no longer occurs, and JAK2, the transphosphorylation cascade, is no longer signaled to control our energy expenditure and our eating habits. So this can lead to uncontrolled eating habits and hence morbid obesity. Now I would like to talk about my genotypic analyses process. Before I could do anything or even start my project, I had to get IRB approval, or Institutional Review Board approval, from the company Advera, is the one I worked with. And this was a very hard task to do just because I had to amend my protocol multiple times, and I was doing human subject research, which IRBs are not very keen on high school students, or really anyone for that matter, doing human subject research. And I'm one of the only high school students to get approval from this specific company. So this has been a major accomplishment for me. But after getting IRB approval, I gathered a cohort of 200 obese pediatric patients from Kids Care Pediatrics in Athens and Madisonville, Tennessee, and I took epithelial cheek swabs from them and air dried them for 2 hours, and after air drying them, I transported them to the lab at my school and extracted them. This would take up to 4 hours just to do a batch of 25, and I did 200 in the end, so that was the most amount of time commitment, was that extraction step. Then after extracting them, I would put them in the fridge, and the next day I would do PCR, which is polymerase chain reaction. This would just basically amplify the DNA into a bulk amount, and I could have a bulk amount to do the further steps. And I would leave this in the fridge, and the next day I would run a gel of these PCR products, and if my products looked clean and the bands were bright, this was an indication to me to move on to the next step, which was the digestion. And the digestion is basically you put in restriction enzymes, which are the scissors of molecular biology, into the PCR products. And this cuts your genome perfectly into the size that you want. And so this restriction enzyme that I used cut the genome into a 416 base pair sequence that included the 223rd base pair where the mutation I was studying occurs. So it cut the genome into that 416 base pair sequence, and basically this was just a more refined version of the genome. And I put it into the fridge, and the next day I ran a gel of the digest. And this was the gel that I analyzed to genotype the patients. Before I started this whole process, my mentor and I ran a gel of our DNA, which is shown in the right here. My DNA is the second in the fifth column, indicated by the arrow, and my mentor, Dr. Abercrombie's DNA, is to the left of that. And given that we had very bright bands, this was an indication to us that we could move on with this aspect of my project. And as you can see, there are 3 more bands to the right of those first 2, and these cleaner bands were a product of us changing the PCR's annealing temperature and kind of moving through trial and error. And so I would say the most important lesson I've learned throughout this whole process is the value of trial and error. Now I would like to talk about my results. At the top right of the slide, you can see a sample gel I've put in here of samples 76, 77, 84, 85, 86, 89, and 90. These are actually the patients that we had to identify as numbers due to HIPAA. But when I look at this gel, I can genotype patients 76 and 77 as heterozygous for the mutation because they have 2 bands, one that is mutated and one that is normal at the 416 base pair. So they're heterozygous, and then patients 84, 85, and 89 are homozygous for the mutation because they're completely missing that 416 band, and they only have the mutated band. 86 and 90 are normal for the mutation. They do not have the mutation at all. So after genotyping 200 patients like I just did, then I got a 7% combined incidence of the mutation, both heterozygous and homozygous, 4% for heterozygous and 3% for homozygous, as seen in the table below. So this was a lot higher than I expected because across the United States, the incidence of this mutation is actually less than 2%, according to a paper that I was modeling throughout this project by the author named Kozrapor. So now the next step is for me to get these results sequence confirmed at the University of Tennessee's Molecular Biology Resource Facility.
So I guess the most difficult thing in my whole research journey has been getting IRB approval, and that in itself was just very difficult, and that showed me like patience is the most important thing in research. Patience, trial and error, like I was talking about before, these are all very important, which I wasn't expecting. I was thinking more like laboratory, something in the lab was going to be the hardest thing, but honestly, due to Dr. Abercrombie's great mentorship, that part was honestly the most exciting and the smoothest. But trial and error, again, that was kind of difficult. But these pictures right here were taken back in February from the first day I worked in the lab, and we've come a long way, and it's just really cool to see now what I can do in the lab. So if you think about clinical applications to the data I've collected so far, if there truly is a high incidence of this genetic mutation in the obese population, then we can further change how we approach the epidemic of obesity and start to include genetic therapy into nutritional therapy and exercise therapy to better mitigate childhood obesity, not just in Tennessee, but in the United States as well. In addition, if there really is a high incidence of this mutation and we find a causal relationship between the mutation and childhood obesity based on further study, we could even add the screening for this mutation into the newborn medical screening. And I'm not sure if everyone knows this, but when you're born, you have to be screened for almost 45 genetic mutations. And so some of those are very minuscule. Some of them, there's 1 in 10,000% chance of you getting that mutation. But if LEPR deficiency, I just got a 7% incidence. Then if we add that to the newborn medical screening, that will be of immense help to the children and their parents. They can further be more aware of what they eat and how much they exercise, and maybe not develop hypertension, not develop type 2 diabetes, and it'll just save more lives. So thank you very much for coming. That was my presentation.